area lead, is the epidemiology therapeutic area lead for immunology and worldwide patient safety at Bristol-Myers Squibb, or BMS. And Doug's been conducting clinical and drug safety research in the pharmaceutical industry for more than a decade across many different clinical development teams, neuroscience, immunology, metabolism, and rare diseases. And he's contributed to numerous successful applications, and his work focuses on leveraging real-world evidence to inform high-quality regulatory decision-making. And if this sounds familiar to you, it should, because this is what pharmacoepidemiologists do, whether you're sitting at Johns Hopkins or the University of North Carolina or Bristol-Myers Squibb. Doug trained at Case Western in the School of Medicine, where he obtained a doctorate in clinical epidemiology, a master's in public health, and I was interested to learn a master's of arts in bioethics. And prior to entering the life sciences, he was an adjunct assistant professor at CWRU, Case Western, and worked in health policy at the Case Comprehensive Cancer Center, as well as the Lewis Stokes Veterans Administration Medical Center in Cleveland. And of many other notable accomplishments, Doug has generously continued to mentor and engage with trainees after having entered the life sciences sector. So internships, practicums, helping to sponsor individuals that are pursuing Masters of Public Health here at Hopkins and elsewhere as well. So as an epidemiologist that has rigorous academic training and who knows the commercial life sciences sector very well, we thought he was an outstanding candidate to participate in this lectureship series. So please join me in welcoming him. And Doug, I'll pass the baton to you.
Great. Thank you so much for the kind words. It's kind of nerve-wracking to hear the way you describe it. My wife called it, I have a career attention deficit disorder that I keep bouncing from places to places. But as things were changing with the NIH funding about 15 years ago, many of us left academia, academic research, which we thought would be our permanent home, right? So now the world is changing even at a faster speed. And even how I function as a pharmacoepidemiologist at BMS also have evolved over the last 8 years. So I thought today, what I can do is, instead of just going straight to a case example, let me just review a few topics. If these are a topic that you have already reviewed outside the seminar setting, please do let me know so I can keep it short and to make sure we can go through all the content within the hour time frame. So just want to at least give you some insight as to how an epidemiologist function within pharmacovigilance or drug safety department in the pharmaceutical department. And at the same time, want to give you some external to a company setting, what are the guiding principle, guiding document that is dictating how or define the role of epidemiologist. Then I will provide a very exciting example on how epidemiologists actually contribute to ongoing FDA discussion around off-label use setting. So we have hoped this example will translate to successful approval for label expansion. And this is a very good example use of real-world evidence in FDA settings. So many of you may have seen this slide before. So if you think of the drug discovery into phases, right? So typically we talk about phase 1, phase 2, phase 3 clinical trial, but in reality, there's a whole lot of other discovery or R&D research activity before you even achieve phase 1 stage. And there could be in the pre-discovery, drug discovery, preclinical development, where you focus on pharmacology, pharmacokinetic research. Along this way, there are many different areas where epidemiologists can actually contribute. And obviously, in the pharmacovigilance setting, right, so our primary focus is around the FDA review and approval process, and also the post-marketing safety monitoring studies. But increasingly, you do find many epidemiologists or pharmacoepidemiologists who are involved or participating in phase 2, phase 3 FDA discussions. At different time, there are different type of studies that we can contribute. For instance, in the early phase, is all around disease epidemiologies, right? So you spend time understanding what is the unmet medical need, what's already out there, and are there any gap in the information? Therefore, can you design the study to fill that gap, enhance our understanding? Once a clinical program get into the phase 2 and phase 3 development space, the focus is more around the target population. Who this drug is developed for? Are there gap in the knowledge around the natural history, disease progression, drug-to-drug interactions, or drug safety? And these are the areas where epidemiologists can actively design and contribute, design the study and lead the data generation activities and fill that gap. So these are the key capabilities, right? As a pharmacoepidemiologist in a drug safety setting, obviously safety is the primary focus. So we do spend more than half our time working with the surveillance team on post-marketing safety surveillance activities. Those include signal detection, refinement, and evolution. So around the signal detection, so we closely follow the FDA Sentinel initiative, and now it's a Sentinel network. Around the evaluation, you think of these as more of the protocol-driven analysis. And obviously, we do a lot on the background rate estimation just to understand, for instance, if we choose one drug or the standard of care as our comparison group in the clinical trial, but once you get to the post-marketing, once the drug is approved, would you do similar type of comparative studies? And if so, were those patients being treated with that drug in the routine clinical care setting, do they have similar or differential experience on safety profile? And obviously, we also collaborate across the functional area with the medical affairs. So these are the external facing part of the company that interact with physicians and prescribers just to better understand what is the research need, what is the unmet medical need, and obviously interact directly with the health authorities such as FDA, or in Europe would be the European medicine agencies. Once the drug is approved, epidemiologists will be the primary lead in terms of conducting or designing and conducting post-marketing safety studies. So while the rest of the development team is focused on getting the drug approved, pharmacovigilance or drug safety usually stay with that program for the next 15 to 20 years, depending by the type of long-term monitoring or observational study we are required to do. Caleb, one question for you. Do you want me to entertain question along the way, or do you want to wait until the end? What's the typical format?
Well, people's preference varies, and either way is fine. I certainly encourage people to post questions. I guess one thing we could do is I could monitor the chat forum, and if I see questions, then I could monitor them and let you know.
Okay.
So, but by all means, folks that are listening, please feel free to engage through the chat forum prior to the end of Doug's comments.
Great. It's simply because I cannot see the chat box.
Of course, yeah. No, I'll monitor that for you.
And
so far, nothing too zesty so far, but
nothing.
But I think these are not highly, but these are really important areas. And as academics, I don't think that we necessarily get a good view of inside of a pharma company and sort of understand the different places where epidemiology is so vital. So this is spot on, and I'll let you keep going.
Great. Originally, this type of talk, I always wish it to be more interactive. Obviously, we are not in person, so I can't gauge the audience reactions. But so please do feel free.
I'll read the room. I'll read the room for you.
Okay,
and we're going to have you back in 2021, and we're going to all have a proper seminar face to face.
Yeah, that would be great. Okay, let me continue then. Okay, so people talk about, so exactly what is the main purpose of drug safety, right? So a different company, epidemiologists or pharmacoepidemiologists could be organized in different ways. For instance, at Merck, they have epidemiologists along with the biostats people. And at GSK, epidemiologists is a standalone functional area, where at other company like Pfizer, then you have epidemiologists split between the commercial aspect, the health outcome research, or the drug safety, but now they have another group, right? So but regardless where you are, what department you're sitting, or who you report to, there are key activity that is universal across different company. It's all around how we plan to describe certain safety profile in the post-approval setting. Everything we do prior to that, you can think of is preparation for the day when we have to design and execute a safety monitoring study. And the legislation that we follow is really around the European Medicine Agency guideline on the GPV, the GVP, the Good Pharmacovigilance Practices. And FDA may have very similar guidance as well. And I will let your next speaker, the FDA speaker, to speak to that. But because each company has to prepare a risk management plan. So that is a document that summarize our position on different risks that we observe during the clinical trial, or risk that is known within that class of medication or known with that particular disease populations. So as part of the drug review process, we have to submit a document called risk management plan that highlight our intent and our planned activity once the drug is approved, to reassure health authority that we have a good understanding of the efficacy profile. We may have some preliminary understanding of safety, but now once the drug is approved, this is how we're going to describe or further understand the risk. And the risk can be classified into different categories, right? There are the identified risk and potential risk. Identified risks are actual safety observation that occurred during a clinical trial, and that's actually unique to that clinical trial. Potential risks are more at the class level, right? Everybody taking this medication for this disease or similar mechanism action or similar class of medication, they all have this as a potential risk, right? So you can go into an argument as a potential or theoretical, but for here we are focused on the potential risk. That means that, for instance, maybe a patient with autoimmune disease, they are a higher risk of developing infections. But then the rate of infection in your clinical trial may not be above what's known in this disease population. But since infection is a known risk within autoimmune conditions, so then you can say infection or serious infection as a potential risk. And obviously there's a key aspect, and this gets to the trial inclusion exclusion criteria. During the clinical trial, there are categories of patients that are often excluded from the study. For instance, elderlies, because they may be concerned that they have a different level of metabolism on the drug, so they may have renal issues on clearing the drug. So elderly over age of 65 may not be included in the study, or patient with pregnancy or childbearing potential. That's another category of patient that's often excluded, or patient with high level or high degree of comorbidity. That's also often excluded. But once the drug is approved, yes, FDA and EMA will provide guidance on how the drug should be used, but it does not always mean that physicians follow that, right? So there might be unique circumstances of prescriber or physician feel like a certain drug will be beneficial to a patient, and they could prescribe in that setting. So missing information could be several categories. It could be elderly population, could be pediatric, It could be women of childbearing potential or patient with high burdens of comorbidities. Right? So when we put in the risk management plan on missing information, it's also a declaration that this company will design additional study to describe this risk in these categories of patients. So the next few slides are just a quick highlight of, when epidemiologists sit in clinical trial meetings, what are the questions that we think, we are thinking about in the back of our mind? Because this gives us an idea that what additional information the team may not be aware of, may not be sensitive to, but these are data points that we can generate as epidemiologists and help the team. We are all about creating the totality of evidence to help the team to make the right decision. So obviously that if you are at that meeting, if you're a core member of that clinical trial development team, these are some of the different questions that we think about and try to see, is there opportunity for us to design the study to support that program? And obviously in the safety, right? So there are different activity that we do, but I think I have gone through quite a few of that already, so we can move on to the next slide. So this is really the overview, right? So before pre-MMA, meaning that before market authorization or FDA approval, there are a set of natural history study that we can think about or design. And then once you decide, okay, we should conduct additional natural history study, there are so many different ways you can design natural history study. Do you want to use your existing big data or the insurance claims database or automated medical records? And in rare disease or specialized disease, you do have opportunity to collaborate outside the company, right, with a patient registry or with a consortium of database. And here I'm just list up some example of some disease registry that could be done by an academic institution, but now increasingly there are data provider or informatic company working with physicians to generate disease registry. So Target Real World Evidence is a company that have multiple disease registry for instance, inflammatory bowel disease, psoriasis, and psoriatic arthritis. We also collaborate with health systems that are HMO-like. For instance, Kaiser Permanente is a closed system. If you are, if you subscribe to that plan, all your care is captured in the same system. Another unique entity is such as Rutgers Health. Over the last 5 years, they've been busy merging and combining different hospitals within New Jersey into Rutgers Health. So majority of the patient in New Jersey can be captured in a healthcare system from end to end of the state. So that's another data warehouse where you have a unique opportunity to really follow a patient, not only across different geographic area, but if they seek care from different hospitals within New Jersey area, that could be a part of that. Right? But at the end of the day, these are all the different tools that we can use to back to the primary purpose why doing it, right, is around natural history. But once a drug is approved, then it gets really interesting because there are so many different functional areas in a drug company that also responsible for a different aspect of the safety monitoring or risk management. For instance, if your drug is has a high potential high risk or different or high event rate of a certain risk, there might be risk management plan that's negotiated, approved with FDA, because obviously the risk benefit profile is favorable. That's why the FDA will approve it. But because of the risk profile, there are additional activity a company or pharmaceutical company is required to do to ensure that the physicians are using it in the safe fashion, right? So those are the risk management or risk mitigation activities. And how you measure the success of those programs, there are different ways to measure it. And oftentimes epidemiology is another good resource to help them to see how well those programs are doing. And we do that by sharing outcome information. For instance, we describe the observed risk in the post-approval setting. And based on the risk, the level of magnitude of risk, they can decide, they can make the assessment whether they're physician education or patient education is working or doing what it's supposed to do. Any question on this very complicated slide that interacting with many functional areas? All right.
Doug,
Yep.
two questions have come up so far. So one of them has to do with heterogeneity of treatment effects and safety,
Yep.
and just kind of how you guys think about that as you are, how you think about that as you're in the early phase of a new product.
Yep.
And then a second question, well, Ayobami, why don't you ask the second question and Doug can hear from you. So this,
maybe I can answer the first question first.
Yeah.
So we do understand that in the real world setting, there will be the patient population is not as homogeneous as a clinical trial. So we do spend fair amount of time thinking about how we would design the study. Will we design the study using intent to treat? Do we need comparison group? If so, what are the reasonable comparison group to design? And then do you analyze the exposure and outcome at the patient level or at the treatment episode level? For instance, in rheumatoid arthritis, patients have a variety of treatment options. They do switch back and forth, either through the route administration or because there's a new drug approved, right? So patients do go back and forth. For some of the risk, that's common in immunology would be long-term malignancies, because it takes time for onset of malignancy. So therefore, how do you assign risk? What exposure do you attribute the risk to? Right. That is a never-ending debate on how do you describe risk, how do you model risk, and then so yes, we do spend a fair amount of time thinking about it. So that, for instance, if I say like next year we have a drug that's going in for a new drug applications. As part of the application, we have to propose several study on how we're going to compare risk or describe risk. Usually it takes about 2 years of thinking and planning and experimenting to figure out how exactly what is the optimum study design, what is the right covariates included in the propensity score model or other type marginal structure model, and then also, what are the comparison group, right? So we do invest a lot of time before, almost in parallel to a clinical program, as you conduct a study, we are thinking of phase 4 planning. Right? Unfortunately, the amount of time needed to design a good study, it still takes about a year or two. Just because it's in pharmaceutical company, it still doesn't happen overnight. It still takes time to do it. Even though most of my end users think that, oh, can you design a safety study, and then want it by 2 weeks from now? I mean, that just right. So it's not a service. It's not like a pre-packaged material ready to go. It does take a lot of time, especially we're in a very complicated disease area that does take a lot of time to think through it, and you also need a disease area exposure as well.
Okay. And then the other question was about HTE, but in a different twist, which is the representation of different groups, such as racial and ethnic minorities in trials, and how target populations for disease processes are defined, and how the criteria differ from how study populations in trials are defined in general. So I think the sort of general question in terms of representativeness and sort of how you understand the safety of products based on the characteristics of individuals that are being studied.
That's actually a really hot topic at the moment, is around the representation of minority in clinical trial. Obviously, we have a history of distrust, a history of abuse, misuse, and I can speak from the bioethicist background that there's a long history of clinical trial or experiment done in minority, and they're just absolutely unthinkable. Right? And obviously, that built a level of distrust. So as the beginning of this year, I actually started last year, there have been many pharmaceutical companies recognize that this is a huge issue we need to address, especially in this era of racial disparities and equities and social justice. Not only we need to increase the opportunity to participate in new trial, but we also need to build the trust that this is not another avenue to abuse like in the past, right? Otherwise, trial participants are often biased by where the pharmaceutical company is able to recruit hospitals or in countries where there are more patients as ready or more willing to participate in trial, right? So there are different reasons why a hospital will participate in trial or patient will participate in trial. And that does provide some sort of a selection bias at the beginning. Yes, randomized control trial use trial randomization as a way to balance everything. Unfortunately, a lot of these issues are embedded in there and are not often well described, right? So I definitely echo and this totally resonate with me in terms of the racial disparity, access to clinical trial, and that is definitely huge. There's definitely a huge effort across the industry or pharmaceutical industry or life science industry try to close that gap. And
Thanks, Doug.
and then actually my project I'm going to go through is actually an example of how we are trying to address that gap, but in a slightly different perspective. All right, so recognizing that we only have about 20 minutes, Caleb, so should I just skip the next section, the use of real world evidence and regulatory decision? What do you think?
Yeah. I think that would be fine. I mean, again, we can go beyond one, but we may lose a few people.
Okay.
So if there are any kind of greatest hits that you think would be great to showcase, you could.
Okay.
I don't want you to feel too constrained, but I also want you to have enough time for treatment of your case study as well.
Sure, of course. So there are a key document that's been published, a workshop in the last couple years. And there's the framework for FDA's real world evidence program. So that showcases FDA's current thinking around how they can incorporate real observational data in the drug approval or decision setting. Duke Margolis has been conducting various workshops on that. And obviously there are new guidance documents coming out both at the FDA, but also at the EMA as well. So let me just go through this example. This is actually a pretty interesting example, and I really hope by next spring, we will be ready to announce the results because this is actually quite exciting. I hope that at the end of this presentation, you guys will be as excited as I am about this program. So first of all, let me clarify. So the drug that we are analyzing here is abatacept. The commercial name is called Orencia. So currently it's approved for patients in rheumatoid arthritis, psoriatic arthritis. There is a sister compound that BMS also discovered is called belatacept. So there are differences in two amino acids, but belatacept has been used in patients with a renal transplant, and abatacept has been used as a disease modulating drug for RA patients. So there are definitely some immune activities, right, for these class of medications. So why are we doing this program? So first of all, let me just go through the importance of the bone marrow transplant. Right? So there are different disease that can be treated using bone marrow transplant as a therapeutic options. There are many leukemia, lymphoma, acute lymphocytic leukemias, and different type of syndrome that can be cured by bone marrow transplant. In terms of the source of the stem cell, they are the bone marrow. You can get it from peripheral blood or umbilical cord blood. But post-transplant, similar to solid organ transplant, they are risk, right? The risk could be dependent by the patient characteristic, but also by the donor characteristic as well. So the overall goal for post-transplant is to prevent graft rejections or graft versus host disease. And the primary mechanism actually is to inhibit both the donor and recipient T cells. And currently, there are a range of standard prophylaxis That's commonly used in solid organ transplant as well. For instance, tacrolimus and methotrexate. And obviously, different transplant center may have their own different transplant protocol, and they do experiment with different dosage. But typically, the goal is to make sure a patient to prevent any graft versus host disease within the first 6 months or day 180 post-transplant. And here's a bit the consequence of current when you do the donor and recipient match. There are certain HLA characteristics. Ideally, we would like to have them match on the 8 of 8 level, but oftentimes you have no choice, but you have to go into 7 of 8 settings. Here, it's just to describe the overall grim nature of bone marrow transplant. Majority of the patients do not survive 1 year post-transplant, even if they have perfectly matched or 8 of 8 match. And as you can see here, the overall survival at 5 years is only about 37% in one patient population and 29% in another. Overall survival is another key indicator as well. So the public health impact, right? So getting data from the National Bone Marrow Donor Program, they've been getting, they've been receiving increasing amount of bone search requests. So this is a formal request for the bone marrow donor program to identify a potential donor for every recipient. In most recent calendar year, they received over 16,000 formal requests, but unfortunately, they could only identify over 6,000 donors and 6,188 transplants occurred. The key issue is inability to identify suitable donors, right? Because 8 of 8 is the allele is the most ideal match and with a slightly better survival outcome. But once you go into other area, then other donor type, the mortality outcome is worse. As it turned out, only 36% of the U.S. population they can find a suitable donor. Actually, sorry, do not have a suitable donor. Right. Let me just go to the next graph. It's very quicker. So here is the really the goal, right? It's really to increase the donor pool because Caucasian right now, the likelihood of finding a match is around 70%. But as you go through the other ethnic type or ethnic group, some are as low as 20%, right? So that's mean for every African-American patient submit a donor request, they only have a 20% chance of finding a donor in the reasonable time frame. And then they sit on the waiting list until a suitable donor comes along. But while at the same time, the mortality rate is at 50% at 6 months or even less at 1 year. So the idea is that if we can safely manage graft versus host disease and reduce the number of rejections in a less than ideal match, the 7 of 8 settings. So look at the red bar. We can actually significantly fill the gap, right, between the Caucasians, the African Americans, and other, right? So this is where the part I think that if we can find a drug that's safe to use in this setting, you can totally democratize the whole process and make sure every patient have a choice, have a chance to have a good survival by 1 year post-transplant. So why abatacept, right? So recognizing time, I'll go a little bit faster here, is that they are, based on the mechanism of action, the way we block the co-stimulatory pathway, it's also could be useful in prevention of graft versus host disease. So there's a phase 2 study, clinical trial, just finished at Boston Children's. So they identified 2 group of patient population, 8 of 8 HLA match and 7 of 8. At the beginning of the trial, both patient group had a randomized double-blind comparison group. But with the emerging understanding of the risk profile in the 7 of 8 population, many ethical committee decided it's actually not ethical or not feasible to randomize patient in the 7 of 8 setting. So the study actually changed the design and they used an external comparator cohort from the bone marrow transplant registry. In the US, every patient going through the bone marrow donor program every center are obligated to share the baseline characteristic for coordination purposes, but they are also required to provide follow-up outcome. So the clinical trial team at Boston Children's thought this is a reasonable external comparison to use to do the study. So what get everybody extremely excited is that patients treated with abatacept for the prevention of GVHD had a significant reduction in the severe outcomes. So the red bar on the 7 of 8 setting is the cumulative incidence of acute graft versus host. And as you can see in the 8 of 8, the treatment effect not as dramatic, but in the 7 of 8, there's a dramatic difference between the current standard of care using external control versus the abatacept group. And if you look at the overall survival, it's pretty impressive. At 1 year, close to 90% of abatacept treated patients were alive versus only 57% in the standard care group. So this is where the BMS got involved, working with the clinical trial team to see, can we further push this to get FDA approval? Here, it just to showcase a different type of mechanism you can interact with the FDA to design the clinical trial and to seek their guidance on the clinical program. So for this particular program, we have many FDA interactions just to understand what is FDA's guidance on how to further develop this drug in this particular usage setting. So this is where the epidemiologists came in. So in June of last year, a group of us went down to FDA to discuss at the end of the phase 2 program, how can we design the next study? FDA actually came back with lots of concerns because they say that we have always asked BMS to do a much larger clinical trial. So why are you coming back with the same study design, with the same study and use of external comparison? More specifically, they had many concerns on how the control group was designed and the method we use to analyze that cohort. So what's different here is, as part of around the same time, FDA has been out there advertising the use of real-world evidence. And the director for that program from the Office of Medical Policy had presented at many workshops on how pharmaceutical company can consider use of observational data. So for this particular study, we actually invite subject matter experts from the Office of Medical Policy to join their clinical and stats review division at this particular type C meetings. So during the meeting is a really heavy stats and method discussion. It's all around how FDA was not comfortable with how the study was designed and how the control group was constructed. And they say that, if we want to come back, if you want to further develop this drug, we strongly recommend you come back with another randomized control trial. And we argue that randomized control trial may not be feasible, but then the physician at the FDA thought there's actually a new regimen being used. Why couldn't BMS consider use of PT-Psy as a more contemporaneous control group? And since they are early signal to show that this actually efficacious. And we actually push back and say, well, use of PT-Psy may be exciting, but it has not been recognized as a standard of care. So basically you are using two experimental treatment regimen and try to compare with each other. And that may not be the most ideal setting either. So what FDA have issue with is how we choose the control, right? The way they choose control is variable ratio match, meaning that some control can be used multiple times, and not every case or not every abatacept patient will have 4 controls. And this actually make the assessment of how well the two treatment groups are balanced, right? So are they similar or very similar? What are the heterogeneity of the treatment population? Is this method is adequate? Because they use a variable ratio match, it made their assessment extremely difficult. And because FDA did not have a chance to look at the phase 2 protocol before the start of the study, so they were not very happy about it at all. And we did do a pattern of use analysis to demonstrate to the FDA that yes, PT site may be what they're interested in, but look at the treatment pattern. Most of the abatacept treated patients are in 2001 and 2015 timeframe. So if we use a newer comparison group, are we introducing temporal bias into the study as well, right? So we did have multiple discussion on this particular time point. At the same time, we remind FDA that in their own guidance document on rare disease, there are criteria that they have suggest if we can meet those criteria, the use of external control should be appropriate. For instance, there's a document on medical need, there's a well-described disease course, and there's a large treatment effect, right? And we also went through our assessment of fit for purpose to using the registry data. So basically, we use that particular type C meeting to remind FDA that, using their own guidance document to remind them use of registry data is totally appropriate in this particular disease setting. And to our surprise, because at that meeting, it ended without a decision. They could not give us the formal guidance because they did not have a chance to talk to the subject matter expert from the Office of Medical Policy, their expert on real world evidence. But they did promise that they will have another meeting. So to our pleasant surprise, after that meeting, they came back and recognized, yes, it's appropriate to use a registry study. But the FDA would like to co-develop that study with us. So we have gone through 3 rounds of protocol revision with the FDA to get this study started. The challenge here is FDA had recommend use of propensity score as a way to address a heterogeneity issue between the 2 treatment groups. But looking at the pattern of use study, it became very clear that we are talking about extremely small sample size settings. Yes, we have a much larger pool of controls, but usually when you go beyond 3-to-1 match or 3-to-1 ratio, you are not really gaining any additional efficiencies in your comparison, your estimates. So basically, we are doing an observational study looking at 50 patient compared to 150, and then we have to use a small sample size setting and find the appropriate methodology to address the heterogeneity issues. I actually borrowed this slide from your previous presenter, had to show that there are different approach to propensity score. Traditionally, for longest time, people used a different ratio or matching. But in recent years, they had taken a more marginal structure approach using the inverse probability treatment way. In the small sample size setting, there are definitely there are key attributes that actually make a very attractive design choice, especially around preservation of the sample size and ability to maximize the precision of estimates. So for this particular study, we use IPTW, but as a supplemental and sensitivity analysis, we propose a matching just to see if we analyze a multiple different way, will we come back with a similar findings. So in the protocol, right, we also emphasize to FDA that yes, when they suggest we use the registry data, if they have the impression that we will have a much larger sample size, we try to mitigate that concern, mitigate that risk, because when you have a limited sample size, in order to do a good propensity score and include all the patient characteristics that could be predicted of 6 months or 1 year survival, there might be many of them. And as it turned out that if we design a study without any regard of sample size, you can include up to 12 or 13 covariates. But the problem is that in a small sample size setting, the likelihood of overfitting is actually become really real. And as it turned out, as we start the analysis, we quickly realized there are collinearity issues, there are quasi-separation of propensity score model. We'll have to quickly reduce the number of variables into the analysis. And also, we did use the inverse probability treatment way to account for the bias and confounding. So we have to explain to FDA how we're going to benchmark how well the propensity score is is performing. And unlike other clinical trial where you can use the observed survival, we actually use the weighted survival, weighted Kaplan-Meier and weighted Lockeran test to demonstrate overall survival. The other key interest here is that in the clinical trial, we look at graft versus host disease, and that could be subjected depending by the transplant center and depending by the amount of information available. Physician may grade graft versus host differently. FDA recognized that. And they also recognize that patient in the clinical trial may have more enhanced follow-up compared to a patient in the bone marrow transplant registry. But we agree that if we use overall survival, that is a very objective measure, right? It's less likely you're going to debate whether the patient had the outcome or not. So they agree that for us to use overall survival as a proxy for graft versus host disease in this population. And the other unique part is that FDA has some concern around how data is collected in clinical trial versus a registry. So for this analysis, they want us to use only information from the registry study. That also creates some opportunity for them to benchmark how well the registry is working, because over 30 or 40 patients actually participate in the clinical trial, and their information will share with the bone marrow transplant registry as well, because that's their reporting requirement. So FDA is actually asking us to do a sensitivity analysis just to see the concordance between information captured in the clinical trial and information entered into the registry study. So this is really the summary of our study and the component that will go into the FDA filing. We have an efficacy study looking at the 7 or 8 matched population using a study design and comparison group very similar to the initial phase 2 clinical study. FDA did not allow us to use the same approach to analyze efficacy in 8 of 8. Their main rationale is a randomized control study was feasible. Therefore, it's inappropriate for us to substitute that with the observational study. So they will only consider data from the observational study. In order for them to gain confidence on the clinical trial or the registry study, they asked us to do the supplemental method evaluation to look at concordance between the two. We hope by the end of the analysis, everything will be positive and we can, the treatment effect is further confirmed, and we will look for FDA filing next summer. I know I kind of went through quite a lot of information, but I just want to at least acknowledge that in order to get this observational study going, there's a large team, both at BMS, from stats, from regulatory, from clinical trial, from programming, to help us to think through every study scenario to come up with the most ideal setting. And we also have participants from key investigator from Boston Children's and also from the bone marrow transplant registry as well. It does really take a small city just to push this program to the finish line. So that's most of the content I would like to share today. And obviously I'm happy to answer any questions. And I know, sorry, I only leave you 4 minutes to do that.
No, this is great, Doug. And then we will, if we can, we'll take the liberty of decompressing a little and
Sure.
maybe going a bit over as we typically go as long as 1:15. Okay. There were 1 or 2 questions in the chat that I can pose to you, and then I think you'll probably have a few more as well from participants. So one sort of that's top of mind is what makes the trials infeasible or not. And is this sample size driven alone, or are there other factors that are guiding the determination that a trial is feasible in one instance and infeasible in the other? I mean, what other factors other than not enough patients makes a trial infeasible?
The feasibility is around the ethical nature of randomized patient to the placebo, because by the start of the study, there are other observations suggest that patient in the 7 of 8 transplant setting will follow an overall survival very similar to the blue line projected on this curve. So therefore, most centers believe that similar to oncology study, if you know your drug works, oftentimes you use a single arm with an external comparator, They didn't feel like it's appropriate to randomize patient knowing there's a 50% or survival by 1 year or right? So it's really the ethical nature of it. And many transplant centers did not feel comfortable contributing patient to the study. So there was a
And
huge gap in terms of how many centers are willing to participate in this part of the study.
okay, and let me just ask a naive question: is that there was no active comparator that could be used internally? In other words, randomized between two different products.
So if you look at right, so if you look at the pattern of use analysis, so majority of the patients are treated with CNI plus methotrexate, right, alone or with other regimens. So that is really the predominant therapy. And so in the original design of the clinical trial, we had the same treatment arm as a comparator, and we had significant issues that most transplant patients would not put them in the placebo. Placebo is CNI plus methotrexate, right? So that is the
Oh, I
commonly
see.
prescribed, right? So the so-called placebo is really the standard of care or the most commonly prescribed prophylaxis for GVHD.
Hmm.
And because of the overwhelming poor survival, and that's definitely create a enrollment challenge, and that's why the study team decided to go back and change the design to open label and take an external comparator from the registry.
Okay. And then, thank you. And then there's questions from Robert Brodkonix and also Lynn Wang. So Robert, are you able to unmute and you want to pose your question? Very faintly, or are you far from the mic or on a headset? We can just barely make you out. Robert, are you with us? Maybe we can go to Lynn Wang and then come back to Robert in a minute. But Lynn, are you with us? Do you want to pose your question? All right. Well, maybe I'll be posing both. So, Robert, I'm happy to. So, Robert was just asking about ethnicity, and when you talk about ethnicity, are you talking about genetically based lines or culturally based identification, or both? And I think here he was referring to non-differential misclassification. So, can you say a little bit about ethnicity and classification or non-differential misclassification?
Yeah, that's actually a very interesting point, right? Because are we saying that if you're looking at the mechanism of action, how we're planning on blocking or modulate the immune response? Are there patient, let's say minority patient, has slightly different expression level on the T cell or the MHC cells or the different receptors? That I have to defer to my genetic epidemiology colleague. But I think here the challenge is that the bone marrow transplant doesn't happen very often, right? So if you look at on over the time of this study, let me go back to the feasibility part. We are not really talking about a lot of patients because too many patients either did not live long enough until a donor could be found, right? So transplant in this setting is relatively small. If I understand genetic epidemiology correctly, you do need a slightly bigger sample size than this to do that sort of differentiation. So for now, when we talk about ethnicity, it's all the disparity in ethnicity is more around access to appropriate donor or access to healthcare system that can help them to identify appropriate donor. Right? So I think that's really so for this particular study, we are addressing mostly the access issue instead of is there any differential expression on the gene profile that maybe their differential response to the standard of care, right? Because I don't know the literature that well to suggest there's such a differentiation, but your point is definitely well taken that maybe other minority, there are other disease where minority have a different risk of GVHD, a different immunomodulator profile. So, but not in this particular setting, at least not to my knowledge. So what we try to
Great.
address is more
Oh,
on
go
the
ahead.
societal level, right, instead of at the genetic level.
Then there's a question from Lin Wang, which is an interesting one about the use of ex-US data. So Lin's question was specifically about registries for rare diseases,
Yep.
but I would be interested for you to speak more broadly about what's the FDA's regulatory position about the use of data outside of the United States for US approvals, or I mean, we know with the conduct of global clinical trials, often pivotal trials, that ex-US data, maybe they want to see at least one trial predominantly US. You could speak to that also, but primarily in the RWE space, how much can you use ex-US data for this work?
It's extremely tricky because every registry, they often fail to disclose the baseline channeling bias or the selection bias. And what makes this bone marrow transplant registry the most appropriate compared to another bone marrow transplant registry that's outside US in Europe is the fact that this registry is a mandatory reporting requirement, right? They are using this registry to coordinate cares and coordinate the search for a donor, but they are by law, they have to do that. And they are also required to report outcome. So your chances of having underreporting or differential willingness to share data with a bone marrow transplant center is, this registry is different. Compared to in EU, the bone marrow transplant is a voluntary data depository, right? So this center set up the infrastructure, but bone marrow transplant, different center can decide whether they want to participate or not. Then you get into then what are we missing there? What are the patients that are not reported into the other registry? Are there any sort of selection bias because of physicians' willingness to participate versus not? Maybe they're just too busy. They cannot keep up the data collection, right? If it's a voluntary, then the standard care takes over, right? Meaning that the daily operation of the care, if they have time, they may report or may not report, right? That takes true dedication versus in the US, by law, they have to do it, right? So there are certain reporting requirement, and that can put the study in the risk. Yes, we can increase this registry to include patients from outside US, but we have some concern that what type of self-selection happening outside US, and do we have enough adequate sample size to address that, right, in this comparison group? So we did go back and forth, and FDA agreed at the end that let's just use the US transplant patients for now. And interestingly, the CIBMTR, the bone marrow transplant center, actually increased the coverage area. And now they actually encourage international participation as well. So this will, going forward, they will have a mixture of patients that are required to provide data versus voluntary, right? So in the future, this registry will evolve as well, right? I think that's just the nature of real world data. They do evolve and they do change. For instance, they are data collection form they are about to implement. That's mean that there are patients already in here, you won't have those data points, right? So they are the level of specificity, level of different mechanism to collect data. You really have to think through all those as you design the study, right? Because the outcome you may be able to analyze in recent years, but you may not be able to use that in earlier year. So if you incorporate patient from earlier year, then you have to reassure FDA that your outcome can be analyzed across all the study window.
What about are there any other settings where you would go to ex-US data for US, label extensions or supplemental NDAs? I mean, if not registry data, are there other settings where you'd use like GPRD in the UK or some, database from Southeast Asia or elsewhere?
So I think the challenge there is some of these drugs are infusion drugs. So data set like CPRD are great at capturing prescription that's managed by primary care physicians in outpatient setting. When you go into the hospital-based specialist, then that's where all the inpatient pharmacy records are a black hole, right? Yes, we can push out a survey to get those data, but then you are at the mercy of who responds to your survey for additional data and who would not, right? So we definitely have considered that as well. And but for now, FDA was okay with using this for this particular setting, but we have considered other medical chart database outside US for other type, right? So we may have our own consortium, meaning we would do a federated analysis. We push out the same analysis plan across 5 to 10 different data partner, allow them to do modification based on what each data partner can or cannot capture. So we have those type of analysis as well. And now the challenge would be, how do you meta-analyze? Because sharing of patient level data across Europe is often prohibited due to privacy concern. So you have no choice but to use meta-analysis to synthesize all the data across different data partners.
Okay, helpful. And then you mentioned that these indications are for acute graft versus host. Is that right? So is chronic a completely different beast? Is there any relevance of any of this to chronic, or does multiple acute give you chronic, or is chronic just not something that's sort of relevant for this product or this conversation?
It is a relevant conversation, but it's all around timing of event. Acute GVHD often occur within the first 6 months, versus chronic can occur anytime after that. So it's also a time window consideration. Right? So we right now we're focused on acute, just because we can demonstrate the treatment effect more efficiently using a small sample size. Once you go into chronic, then there's a whole different sample size and time window considerations, right? So we haven't gone that far yet. So if you think about it, I mean, you have the universal GVHD we try to prevent, but we focus on the small area first. When we can demonstrate this effective, then you can slowly expand to other grade or severity of graft versus host.
Right.
So it's very similar to oncology, where you focus on third line who have failed everything else. If it work, and you slowly work your way up to primary or first line therapy.
Okay. Great, Doug. Are there any other questions for Doug?
Yes, this is CG Wang. Can you hear me?
Yes.
Yeah, we hear you great.
Thank you, Dr. Cole. This is really a wonderful presentation. The case study you shared with us is very, very informative and helpful for us who work on this topic. So I would like to ask you a question regarding one of the specific concerns FDA raised at one of your first meetings with them. That's the concern about the definition of the endpoint. I think you listed it maybe on slide 28 or something. But there seems to be this difference between the definition of the outcome on the clinical study side and on the registry side. So that's when we use propensity score methods to adjust for so-called covariates or patient characteristics, and we can only, my understanding is that we can only adjust for those patient level covariates. But when it comes to this group level, in this case, arm level covariate, namely the definition of the outcomes, propensity score method probably won't do much for us. Through my interactions and collaborations with the medical device center statisticians, I get the impression that they encourage sponsors to downweight or discount the amount of information from the real world data sources, even after you get a matched or selected patients based on score methods. I'm wondering, in this particular case study, did the FDA, how was this concern about the differences in the definition of endpoints addressed, and whether FDA, in this case is the CDER, of course, suggested anything related to this so-called downweighting external data sources strategy? Thank you.
Yeah, so great. So their proposal was in the meeting was very casually say, oh, why don't you look into propensity score as a mechanism to address bias between the group. And that was the only time they mentioned it. And they did not provide any additional feedback on that. So there are two issues that you brought up. And what's actually very, very close to this topic is around outcome definition. In the clinical trial, acute GVHD was evaluated at weekly basis, meaning that every week, the center will ask the patient or ask the physician, did this patient have acute GVHD? Yes or no? All right, they will ask on a weekly basis. At the registry, it's at a 6-month level. Or did the patient in this last 6 months, did that patient have acute GVHD? Right? So there's surveillance issues. Obviously, ideally, both data collection method will end up with the same information, right? Because if it's acute GVHD, it shouldn't matter which way you collect it. You should get to the same endpoint. I mean, the same information. But FDA have concern because clinical trial is assessed at a more frequent basis, and versus CIBMTR registry is based on whenever the center provide at 6-month time point. There might be some data resolution issues, right? So that's why they say, okay, then let's look at only data from the registry. And so they think that there are two benefits to that. One is maybe the registry had additional patient that was not part of the trial, that may be in an off-label use setting, but if the outcome are analyzed or collected in the same fashion, the same mechanism, then you don't have to worry about the differential or surveillance bias between the two different groups, right? In terms of propensity score, there are two issues there. One is, if you recall, the treatment group, at the end, we can only identify about 50 patients. So most of the propensity score have provide guidance almost like for every outcome, you need to have certain number, for every outcome, you need a certain number of patients with that outcome in order to analyze. And they have a minimum threshold that you need to have at least 100 outcomes, right? So the predicted outcome here is patient being probably are treated with abatacept. We only have 50. So when you look through the long list of patient characteristics and the many categories that can be in each variable, we quickly run into issue where we have many controls with that response, but the case did not. But then if we say, okay, then let's provide additional filter to choose our comparison group to make sure we have that. But then FDA came back with, make sure you pick a control that's representative of the entire registry, right? So you can't really apply filter to make sure you have maximum match to the abatacept group, but at the same time generalizable to the entire population. So we settle with, let's randomly select 150 control patients with 1 or 2 filters, and then let the rest sort itself out through propensity score model. Right? So that's the best way to. It's a compromise between maximize the performance of propensity score and to make sure it's generalizable to the overall transplant population. Right? So that was the two key issue that we kind of went back and forth and 2 teleconference just to discuss that point.
Great. Well, Doug, you've made us better epidemiologists. So thank you again for visiting with us and providing this outstanding overview, not only of the life of an epidemiologist inside a big pharma company, but also a very nice applied case study that let us dig into the details a little bit. So thank you to all who joined us, those posing questions, those that stayed to hear, and we wish you the best for the holidays if we don't see you tomorrow for a coffee hour at 8:30 in the morning. And Doug, we also want to give you 15 minutes or so break. Thank you for your willingness to meet with some trainees at 1:30. And Jody, anything I'm missing that we should say?
No, that was terrific, Doug. Thank you.
Thank you, and thank you for the opportunity to share some war stories. I don't have many new ideas, but I definitely have many battle scars with the FDA. Right? So I don't think from our lesson learned, maybe there's something new you can learn from that. So thank you for the opportunity to share war stories with you. Thank you.
Yeah, it was outstanding, and thanks to Jamie, and thanks to all. And this marks the close of this lecture. So thanks again.